Good afternoon, everyone, and it's my great pleasure to be here, present our research, and thank you for your attendance, first of all. So this is a 1-hour talk. Today I'm going to cover basically two topics related to nanoplasmonic enhanced bioimaging. The first topic would be, of course, let me start my talk by telling you why we're doing the actual research. What's our motivation doing the research? Okay. Our research motivation is, it's very different from many other people. So we're trying to inventing a better equipment or better optical system to do a better imaging with higher resolution, higher sensitivity. We actually approach this problem from different angles. We want to make things cheaper, but better in terms of performance. So the status quo in the molecular cell imaging is usually you have to use very high cost. Of course, sometimes they're high performance, not necessarily, but they're usually high performance systems. But it's very expensive and very bulky and benchtop. You probably can find many equipment like that in this building, in Beckman, probably in a basement or first floor. You can find many state-of-the-art optical imaging systems that can do a very good high performance imaging, but they're bulky and very expensive. So they're not quite affordable to the application, especially requires low cost and portabilities. Our goal is to develop, using micro nanotechnology, to develop a low cost but high performance systems, and can do something equivalent to that expensive system, but sometimes can even do a better job. And our main application is to have this portable devices to allow people to use in a bedside or at the point of care, for example, in a doctor's office, right? And so that will also emphasize on the high affordability. So then many people can enjoy the high affordability to use that for portable applications. So overview is here. Today I'm going to talk about two aspects of this nanoplasmonic enhanced imaging, bioimaging. So they are, the first one is colorimetric plasma resonance sensing of biomolecules using a device we developed. It's called nanoelectric copper ray. The second one would be using nanoplasmonic substrate to enhance the sensitivity of three-dimensional fluorescence confocal imaging of live cells. All right, let me start from the first one, but before I talk about that, let me just give a very brief review. Definitely, this is not a lecture about plasmonics. There's really only have 2 slides about plasmonics fundamentals. So so-called plasmonics basically is a coherent oscillation or collective oscillation of free electrons in the metals, especially at the interface of the metal and the dielectric medium. So you can basically excite the free electron oscillations in either metal film or nanoparticles of metal by illuminating light on the surface. But there's an intrinsic problem about the surface of plasma, which is the mismatch of the dispersion curve with the free space propagating electromagnetic waves. So if you look at this, this is basically describe the alternating density of free electrons along the surface of the metal and dielectric interface. So that will form a surface wave, which is plasmonic wave on the surface, on the interface between dielectric and the metal materials. And this, if you look at the dispersion relationship for free space propagating electromagnetic wave and the plasmonic waves, they don't really match that well. This straight line representing the dispersion curve for a free space EM wave, and this red line representing the dispersion curve for a cytoplasm wave. They don't really have overlap in most of the cases. So you have to play some tricks to make these two intersect with each other. So the typical tricks people do is either place this in a high refractive index medium, so then you can further bend down this dispersion curve to make it has sort of intersection point with the dispersion curve of the cytoplasma wave. The other way to do it is you can create periodic structures, so then you can modify the K value here to make it has sort of an intersection with the free space EM waves. But today my focuses on how do we use this plasma resonance effect to do bioimaging or biosensing, right? So people, I mean, over the past 30, 40 years, people already are doing that. And you probably know this very well, SPR, or surface plasma resonance biosensing or bioimaging. It's already become commercially available. And there's a company called Biocore, started about 10 years ago and was acquired by GE Healthcare. So now GE Healthcare is basically own this technology, own this brand for making this SPR sensing equipment to allowing people studying protein-protein interactions, protein-DNA interactions, or drug screening on the surface of a thin film of gold or silver. So the concept is very simple here. You just need to illuminate a light source on the surface of this gold film. And then you use another detector to measure the reflectance. Okay, measure reflectance. Of course, on the other side of this gold film, you could have some biological interaction happens. What it shows here is you can have a molecule layer forming on the surface, or you can also have molecule binding on the surface. That will change the surface plasma resonance property of the film, which can be then measured by measuring this so-called angular spectrum, or the reflection spectrum from different angles. So this is very typical data you measure in the SPR imaging, that you measure the so-called resonance peak positions. In this case, is resonance angle, right? And then and by measuring this angle change, or the resonance wavelength or frequency change, you can then somehow tell the difference of the molecule layers, either due to binding or dissociation of molecules. And so this is a thin film-based technology. And so in the past 10 years, including myself, we're doing this nanoscale plasmonic sensors or imaging tools. And so in that case, we started making nanoparticles of silver, gold, or other metal materials, or using electron beam or FIB technology to nanofabricate some periodic nanoplasmonic structures, including, in this case, what you're showing is SEM pictures of a gold nanodots array that form collectively either green or red colors, depending on the size of the particle you made, depending on the periodicity of the particles in that arrays. So they can be used as a sensor to do a very similar function, very similar sensing as that gold thin film you see from previous slides. Of course, you can really play with a lot of varieties of the structures and to create different kinds of sensors. In this case, it's showing an array of gold nanowires in different width. So from the left, from the right-hand side to the left-hand side, basically, you are seeing the increasing diameter or the thickness of the wire from 20 nanometer all the way to 150 nanometer. And depending on their size, so they're having different resonance frequency or resonance wavelengths. That's why you're seeing different color exhibit in this case. All right, so there's one problem still not being solved. Here is basically you still need a very specialized equipment to do all kinds of sensing. I'll show that. This is another example showing that you can basically not only making spherical particle, so by changing the shape of the particles, you can really tune the color, which means tune the plasma resonance wavelengths of that plasmonic structures, right? Here's from, so we can make from blue all the way to red particles depending on their shape and material and size. And you can also make periodic holes or particles and to collectively exhibit the same color. In this case, H mu was made by punching an array of holes through a gold film, and the transmission line will show up individual colors as H mu, H in red and the mu in green, because they have different pore size and different periodicity. That's why it shows different colors. Okay, so in my lab, what we do is we really want to make this into a engineering effort rather than chemistry efforts, okay, because we're competing with a lot of chemists at the same time. We want to use a very advanced nanomanufacturing technology to make a large area, highly repeatable, highly uniform nanoplasmonic substrate. In this case, we use a technology called a laser interference lithography to make this nanopillar array across the entire 4-inch wafers with very high uniformities. And this can be done also at once because this is based on a parallel lithography technique. It's not like E-beam, you have to do point-by-point scanning. Here, you basically do illumination at once of the wafer. You got all the nanopillar rays at once across the whole wafer surface. All right, as I said, there's still a problem we need to solve, which is equipment problems. The equipment used to measure those nanoplasmonic structures is still very bulky, very complicated. In this case, I'm showing that what's usually being done for measuring a scattering spectrum of a single particles. In that case, you have to use dark field condensers to illuminate light from oblique angles, and then you measure the scattering light from a smaller scattering angles using objective lens. And then you send the light into spectrometer or send the light to an imaging camera to analyze the spectrum of that scattering nanoparticles or plasmonic particles. Okay. And so this example is showing that using that kind of a system, what we can do is we can put in nanoparticles into a living cells and then monitoring the color change of an individual particles within the cells. Of course, all the all the particle here is primarily already internalized by the cells and resides in a cytosol. Okay, resides in a cytosol. Okay, and we also develop something called a whole field high-speed nanoplasmonic imaging systems. Allow you to do a full field or whole field imagings of nanoparticles in your whole field of view at once. You don't have to basically move your stage, so you can get the spectral information of all the particle in this field of view at once. But still, a little bit requires some sophisticated equipment. So we decided to do something better, especially for portable applications. Actually, what also make us motivated in this case is people already demonstrated very high sensitivity in their nanoplasmonic biosensors. For example, Rick Van Dyne's group from Northwestern, they have demonstrated that using just an array of silver nanoparticles, they can show sensitivity as high as hundreds per refractive unit. So this is already very high sensitivity, allow you to even detect the molecule binding event on the single particle surface. So what we decided to do is to do a better job using engineering manufacturing technology. And here shows that we also get some inspiration, some ideas from ancient Romans. I mean, they are really amazing in terms of scientific development. Almost 2,000 years ago, they already figured out a way to make silver nanoparticles, gold nanoparticle in a very uniform size. From 20 nanometer all the way to 200 nanometer, and they can blend the nanoparticles with glass, and as they can start making this glass wine cup, and with a lot of the nanoparticle embedded in there. Because they're making nanoplasmonic particle already 2,000 years ago, so they can really change the color of that wine cup just by changing light illuminations. Now this shows that the cup actually used to being exhibited in Art Institute of Chicago a few months ago, but now it's basically shipped back to British Museum and back to its original place. And the reason why I changed from green to red colors or the red to green is because here the photographer is changing the light illumination from front lighting to ambient lighting. Then you are detecting the absorbance and the scattering peak of these nanoparticles in the glass sidewall. That's why they exhibit different colors. So we learned from that. We think that why don't we make the same cup with basically the nanoparticle decorated on the sidewall of the cup, but we want to make them much, much smaller. So we shrink the cup size for about 1 million times. So it's 1 million times smaller. You're not going to see that by your naked eyes. You will see it by using electron microscope. And then, of course, one small, tiny little cup will not do a lot of good thing for us, especially for naked eye visualization. So we make about billions of them into an array and place them down into a highly ordered forms onto a plastic film. Okay, so this is what we call as a nanolithograph array that consists of about 1 billion of such a nanoscale lithograph. And then what we also trying to demonstrate is by using a similar illumination lighting change conditions, we can change the color from red in this case, we're changing to ambient light. Then if we basically shine a light in the front surface, the color changes into green, as you can imagine. So it turns out to be that it's this world's most sensitive plasmonics sensors so far has been published in the paper. This is really an idea that inspired by the old technology, Asian technology, and make it better using a state-of-the-art manufacturing technologies. So if we compare our nanolithograph array sensor with the conventional SPR sensor, is we really can do a nearly equipment-free imaging and sensing. So before, on the left-hand side, you see a configuration of a typical SPR imaging system. Now is being selling by GE Healthcare. So you need a very sophisticated light source, and the detectors are mounted on a highly controlled rotational stage, so you can control the angle of your light source and your optical acceptors, detectors. And then so they can use that to measure your angular spectrum and to find out that resonance peak. So this is a very sophisticated system. It requires very fine mechanical adjustment. But in our case, the detection becomes so easy. What we actually do is we just use a wire light source. You can get the source from your flashlight or from an LED light source. Only costs $2 each or even cheaper. And then on the other side, you just need to have a color camera. It could be your Nikon digital camera. It could be your cell phone camera. That's it. And then we can do a similar sensing as that equipment and very expensive systems. So what you do is, what we envision is to have something that's equivalently sensitive or even more sensitive than a current bulky system. It costs more than $1,000, sometimes even $500,000 depending on the brand of it. And we want to make something like this on the right-hand side using a cell phone camera as imager, not really anything else. You just need an LED source or even a flashlight you can get from Walmart. And then you can, using cell phone camera, to take image, a colorimetric image of the SPR sensing. So in terms of applications, is how do we make this nanolithograph? Okay, we start with this nanopillar array, as I showed previously. And this is made by using laser interference lithography. So we make this on a whole wafer level, and the periodicity is about 250 nanometer. Center-to-center spacing distance, and the size of the pillar is about 180 nanometer in diameter. The height is controllable by the etching time. So we have the height from 200 nanometer all the way to 1 micron. So this really shows a very high aspect ratio, very long pillars. Then we use that pillar, and this is shorter pillars. This is in a medium size. And then we use this as a mold to do a nano imprinting or nano replications of plastics. So it's really like a hot embossing or injection molding, but do this in the nanoscales. So basically you inject the liquid of a plastic monomer onto the mold, then cure it, then you peel this off, or to separate this mold from the plastic side. So once you've done that, you basically can create a negative geometry onto the plastics. And then after this, we can deposit a thin layer of silver gold onto the surface. And because we are starting from a nanopillar, which is also a slightly tapered nanopillars with some angles in the sidewall. So after replication, we're getting a cup size instead of a hole. Instead of a straight sidewall hole, we're getting a cup with a slanting sidewall. So that allows us to depositing some gold particle, silver particle along the sidewall of that structures. It really form something like a cup structures. You have a nanoparticle on the sidewall, not only on the top surface. Okay, so then we start doing experiments. So we start from, of course, a small piece in nanoscale, this is already a very, very big piece, right? You know that because every individual feature is only hundreds of nanometers. So 1 centimeter by 1 centimeter device is already considered very big. And now we have a wafer scale size of device. So we started doing some experiments by playing with it. And even at the beginning, we started using a cell phone camera as our imaging tools. And now, I mean, here it shows that if you drop just drop a DI water on the surface, the color immediately changes. You can take this color change by using cell phone camera. And of course, if we want to study the microscale color change, we have to use microscope. But that could be the simplest microscope you can buy. So we spent about $2,000 to get a very cheap bright field microscope. That's more than enough for us to take this image right here. It shows that at microscales, there's still some defects here that actually black dots. This is actual defects from manufacturing. This is still not 100% perfect yet. But you can see before adding anything on the top, this shows as a green color in transmission. But once you add water, drop in there, and inside the water it shows as an orange or yellow color. The color already changed. And this, I think, we believe is the first time in the plasma resonance field showing the actual color metric sensing of material. Because before, people always use spectrometer to do the sensing, because either shift is too small or you have too many peaks to see any colors. So here we only have one individual peak in a whole visible wavelength range, and we have large shift. That's why you can see the color change by eyes even. So we can work on this for both reflection mode and transmission mode. So this shows the transmission mode, which is similar to this. And you can also do the reflection imaging, and then the color will be completely complementary to the transmission mode. Okay, in this case, the color in the reflection for the air case is red, and for the water case becomes blue, green, sorry. And then what we do in the following is, why don't we do more fancy stuff, really trying to detect more molecules, more liquid on the surface. So we start to make this palette, basically, by mixing different liquids and mixing different biosolutions on the surface. It shows that glucose solution and sodium chloride solution and the mixture all give very different colors. And this is an air part. Okay. And so then we have also done some very preliminary studies by just putting a DNA solution droplet on the surface to see whether that concentration of DNA in water will affect the colors. It does affect the color, of course. In this case, the DNA concentration has huge difference between this 3 droplet. This one, the concentration is about 100 micromolar. And this is a 100 nanomolar, this is a 100 picomolar DNA solution. If you see the color inside this part, you can see the color really varies from a low to high concentration. So we can also do some extrapolation. We use an algorithm, not to measure the spectrum per se, but really using, by analyzing the color, the RGB value in this image, we can extrapolate, we can basically simulate the actual spectrum of those colors. All right, so then we start to do a much more systematic studies using this device. This shows that we start using and testing many different liquids. It shows air, water, glucose in different concentrations. They will change color depending on the concentration of glucose, and salt solutions, and many organic solvents. Here's chloroform, glycerol, DMSO. They all show into a different color because their optical refractive index, other properties are different from each other's. And we can also do patterning, apparently. So we do a patterning, we pattern this sensing area into a letter patterns, nano or Illinois, and it shows that this sensing really happens locally. So you can have those solvents or liquid drop into a localized positions onto this pattern, and they will show different colors depending on what kind of liquid is being put on top. So as I said, the actual imaging becomes so simple and so equipment independent compared to the commercially available SBR imaging system, which costs a lot of optics in there. Here we need it only for the microscale imaging. We only need a very inexpensive transmission mode or reflection mode bright field imaging microscope to do that work. If you don't care about a microscope, you don't care about micro-scale imaging, you can simply just using your cell phone camera to do the work. So here it shows that we can also use this tool, this sensor, to study fluid dynamics on the surface. This is a video captures when there's two droplets of organic solvents evaporating surface. It shows that on the left shows the ethanol start to evaporating from the surface, so the colors return from the red or orange back to its original green or blue colors. This shows the toluene drying. And we're actually using this for studying a lot of fluid dynamics without adding any dye or molecules in there. Okay, so of course, to do a more quantitative analysis, we want to compare our color analysis, which is RGB analysis, with the actual spectral measurement. That's conventional quantification methods. So we compare these two methods, head to head, and find out that we can use this RGB analysis to differentiate different liquids, just depending on what red, green, and blue channel intensity, and we can categorize those organic solvents and liquid solutions into different categories. And after categorization, of course, we have asked ourselves why we're seeing such a big change in color, and why we have such a high sensitivity. So we start looking at the into the nanostructures. What this special nanostructure make this sensor so special? So what we find out is, so this kind of a geometry and the shape and the material give us a single resonance wavelength peak in the whole visible range. That's the key to give you a color sensing, a color metric sensing. Let's say if you have all 7 colors, you have 7 peaks in your spectrum, you're not going to see color. You're going to see white light, right? So you're not going to use that. Even if that could be very sensitive, you're not going to use that for colorimetric sensing if you have too many peaks. So if you want to do colorimetric sensing, you have to only have 1 peak, at most 2, in your spectrum. So that's what we have accomplished. And most of other sensors so far, SPR sensor, they have multiple peaks in the visible range. So they're not be able to do colorimetric sensing. And also, this thing can give a much higher sensitivity. Of course, the sensitivity varies depending on the refract index range. Especially for the refract index, very close to the refract index of water, 1.33, we have very high sensitivity. This is a sensitivity we measure using sodium chloride solutions. We start from 0.1% of sodium chloride and going up all the way to 20% of the sodium chloride. And we find out, especially low concentration range, we have sensitivity is almost close to 50,000 nanometer per RIU, per refract index unit. That's really a world record in the publication, of course. Maybe the company has even better performance. And the figure of merit is above 1,000. Okay, so with that, we actually want to find out why this particular cup structure, especially the nanoparticle coating on the sidewall, gives such a high sensitivity and such a unique, a single peak resonance spectrum. So this shows that we did some FDTD simulation to compare different geometry, different designs. This is a very typical design. You can read about this in many papers, maybe 1,000 papers published previously. That is just a straight sidewall nano hole structure. You can deposit gold or silver onto the surface, but you don't have anything on the sidewall. So what you get from this kind of geometry, you can have really multiple peaks in your visible range. That gives you blended colors, which becomes white colors eventually. So you're not going to use this kind of sensor for colorimetric sensing. So once you start making this slanted sidewall structure, and especially when you have this particle coating the sidewall, you start seeing a single resonance peak in the visible range. That's very similar to the mist scattering of a single particle. Okay. All right, so and we published a paper as a cover article in Advanced Optical Materials, and it's highly reported in many places like Wire Magazine, Forbes, Smithsonian, because people recognize this could change the way that people do biosensing. I mean, usually you have to spend hundreds of thousands of dollars to do a very sensitive sensing, but now you only need a cell phone camera and LED light source to do this. It does not take too much money and effort to do that. So we, in the paper, we also demonstrate that we can do DNA hybridization detection without any labeling. So it's all done by just colorimetric imaging. So first of all, while this is very, very simple studies, first of all, you put a single strand DNA on the surface. You can conjugate it on the surface. Then you introduce the complementary strands. Let them hybridize to form double-strand DNA. So after hybridization, we can see the color changes, and the color change is proportional to the concentration of the DNA on the surface. Of course, for any sensors, you have to ask yourself, what's the sensitivity and what's the dynamic range? So the dynamic range of the sensor is relatively narrow. It's only basically span about 3 to 4 orders of magnitudes. So the actual most sensitive region for this kind of sensor is around sub-nanomolar and a nanomolar concentration range. For a concentration higher than 100 nanomolar, this sensor starts to saturate. You don't see any color difference. So that's our dynamic range. So for this particular concentration range, this sensor could be very valuable. All right, so then we start looking at other possible application of this kind of SPR sensors. And because in my lab, in our lab, we also do a lot of microfluidics. For those of you who have some experience or studied microfluidics before, that you can easily be fooled by these pictures. But this is a very famous picture and published by Stephen Quick's group at Science 10 years ago. It could be easily fooled by this while you thought, okay, microfluidics could be so colorful, but actually it's not. Microfluidics usually is colorless. You're not going to see any liquid in there because contrast is so low. You will not see even channels. The reason why you're seeing so much, so many colors, because they use food dye. They use blue and green and red food dye to fill the channel. That's why you can see the channels. But usually you're not going to see any channels because the actual refractive index of water is very close to that of the polymer used to make this microfluidics. So we think that we can really make microfluidics to be something colorful, and we can detect molecule and liquid without adding any food dye in there. So the fabrication is very, very straightforward and simple as well. We started making PDMS microfluidic channels and then just permanently bond this PDMS using optical adhesive onto our nano LCA device. And once the bonding is done, you can use it as a colorful microfluidics. And the imaging is also very simple. You just need a halogen lamp or flashlight or LED light source shine from the bottom. Then you can either use a microscope objective lens or without it, just using cell phone camera to take large field image. And what you get eventually is something like this. So this picture is really taken by cell phone. That it shows that we overlaid this 10 parallel microfluidic channels on top of this nano LCA device. Once we feel liquid in there, we actually feel different liquid, but you're not going to see the liquid. The liquid is going to be colorless liquid. The water and ethanol, just by looking at it, you're not going to tell the difference between these two. So we actually feel liquid in there, but you're not going to see them for the area outside the actual nano LCA area. They all look like the same, transparent. But once they feel in the region that is on this sensor area, you start to see different colors showing up. Okay. So now we making microfluidics really into a colorful microfluidics. So this is another picture showing integration of a different design of microfluidics onto this nano LCA device. Okay, and this microscopy picture shows that with and without filling the water in a channel, you see the color change from green and to orange after filling the color. After filling the water in there. And so by combining these two, a very powerful two together, we can really study a lot of things. Like, let's say you want to do chemical reactions. You want to mixing different reagents together. how do you monitor the actual reaction happens? Usually, no matter it's reactor or not, or how much is it, you basically cannot tell any difference in colors. They all look like transparent to us, right? But now, by using this sensor, you can mix them together. The color can tell what's going on the, in an inside of liquid. So here shows that, if you try to imaging the mixtures of water and the glycerol, depending on the concentration, and they can really exhibit different colors. So this one is a pseudo color plot of the intensity at the blue channel, because now we're taking RGB images. So this is a particularly blue channel image. We can also plot a similar image for the red and the green channels. All right, so what we actually eventually want to do is something like this, and we're already built a prototype of that. And this is such a simple systems. Basically, we spent $1 to buy a LED light source. Only costs $1. And of course, the actual machine shop costs more at this moment to build this optic tower. And then we insert this device, this microfluidic nanoscale device in between, and putting a cell phone camera on top. That's it. And we're taking this colorimetric imaging of SPR. Okay. And so this is some repeating slides of that. So here it shows that by taking this color image, you can start analyze their RGB channels to really differentiate different liquids by their intensity in different channels. Now what we can do also is beyond the static imaging, we can also do some dynamic imagings of the microfluidic droplet inside microfluidics. Okay, so here it shows that we're trying to make a water droplet in oil. This is a very popular topic nowadays in microfluidics field, that people wanted making a droplet-based reactions or bioreactors or sensors. So here it shows that we can make water droplet into oil, but people have done that for many years. But before, you cannot really tell any difference because they all look like transparent to us. But now, because oil will show different color than the water on this device, so you can really do a colorimetric imaging of that fluid dynamics. So I probably will do really quick. I think the time is running up. And showing that, okay, this is a field channels and unfilled channels. So what is the most important things is we can do dynamic fluid dynamic imaging by using this colorimetric sensors on surface. So here shows that we making this oil droplet by doing dynamic focusing of water from the side channels. Okay, and of course we can do quantitative analysis of the color of the RGB value to really find out the actual mixing conditions of these two liquid at microscales. Well, imagine that if you are doing chemical synthesis of a drug molecule, a drug compound, you start with a reagent A and reagent B. They're all colorless. They're all transparent. You don't know what happens conventionally. But now, once you mix them, you can use this color change to monitor the actual chemical reactions or biomolecule bindings in there. And of course, we can do some manipulation of the flow and to show this is really response to different materials. Okay, so that leaves me only have probably 25 minutes to finish the second part. The second part is kind of related to the first part, but we want to really couple the plasmonic devices with what most biologists typically do, okay, which is a fluorescence-based confocal imaging. I believe almost nowadays, as long as you are doing cell imaging, you always do some sort of fluorescence confocal imaging. Even in this campus, we can find out probably more than 100 research groups in this campus who are doing fluorescence imaging, some sort of fluorescence imaging. So our goals here is, can we invent a device that can help all these 100 biology labs in U of I campus or 1 million lab all over the world by really making something simple for them to use. They don't have to make any change to their current imaging systems. They can just get this device from us, start doing their cell imaging on the surface of our device. That's the motivation we have. So we wanted to enable by using this device what we call a nanoplasma core, or in the full term is coupled optical resonant excitation, nanoplasma core device to enable three-dimensional fluorescence enhancement for 100 volts. Basically, surface-based fluorescence enhancement for 100 volts is already demonstrated by many groups, but three-dimensional enhancement is really not. Sorry. And our goal is also, without changing any current optical imaging system, you don't want to tell your biology collaborators, you need to change your optical system completely. They don't like that. They want to keep the same system, but getting those benefits. And this also potentially allows capture single molecule events in the living cells, because now you're enhancing this fluorescence sensitivity for 100 volts three-dimensionally. So before you can only detect 100 molecules, now you can detect single molecule. Okay. And can also help to observe very early stage protein expression soon after gene transfection. I'm going to show the example of our work to actually demonstrate that. So the near field or surface-based fluorescence enhancement from metal surface has been demonstrated by many groups, by maybe 1,000 groups all over the world. That's basically nothing new there. People know that fluorescence can be either quenched or enhanced depending on the distance between the fluorophore molecules and the nanostructured metal surfaces. Okay. But this only goes beyond 100 nanometer above the surface. If you're talking about 10 micron above the surface, you don't have any influence in terms of fluorescence intensity from the surface properties. But we want to demonstrate is we can really extend this influence 10 micron or even 20 micron above the surface. How do we do that? Of course, we have to make some assumption here. We have to work with the cells that's almost spherical, almost spheroid at least, and then putting the cell and culture the cell on the surface of this nanoplasma emitter surface. Okay, and then what will happen is the free space EM wave excitation will be coupled into cytoplasma on the substrate, and then eventually coupled into this whistling gathering mode resonance in its cell cavity. So here we view cells. We view cell as a spherical optical cavity to sustain that whistling gathering resonance mode. Okay, this is very bold ideas, and we have already proved that in our simulations and experiments. So here shows in the simulation that you basically can really launch a free space wave, and then through the mediations of this nanoplasmonic substrate, you can excite this whistling gathering mode resonance inside the cavity. I think this is probably the first demonstration. No one have demonstrated this kind of thing yet, because you always need an optical fiber or something like that, or waveguide to couple this free space light into the whistling gathering mode of the resonant cavity. But now we don't need to do that. You just need to have a substrate, a plasmonic substrate, and you can use a free space EM wave to excite that whistling gathering mode resonance. Okay, so because of time limit, I will just probably skip some of this simulation result. We have done a very systematic simulation result to vary the size, vary the periodicity of the plasmonic structures, and also vary the size of this cell diameters to see what's the coupling properties between these two. So this is really SPR to WGM coupling effect. And we then also did some semi-analytical computations to find out what's the coupling efficiency or coupling conditions between SPR and WGM. If you're interested, you can go to our paper to look at the details. But just let me show you the experimental aspect of this. How do we use this kind of substrate to enhance fluorescent intensity of the cell for 100 times in a three-dimensional space? So here what we did is we make something similar to our design, but not completely yet, because we want to make some low-cost substrate for people to use for many times. So instead of making a highly ordered nanopyramid array or nanocone array, we started making some semi-random nanocone arrays, and then started depositing gold or silver on the surface. The benefit of this, of course, we actually have some benefit enjoying from this randomness is we're creating a broadband plasma resonance substrate. Instead of having very sharp plasma resonance peak or modes, we're having a very broad mode in a visible light range. So then you can always find some overlap of the resonance mode with the fluorescence, excitation, and emission wavelength. Okay. All right. So this shows that you can also provide some near field electromagnetic field enhancement. Okay. And then, I mean, this work shows that if you put a molecule, fluorescent molecule on the surface of those nanoplasmonic structures, you can see the enhancement of fluorescence on the surface. But this has been observed by many other groups. And this shows some characterization of plasma resonance using patholuminescence and electron microscope. Okay. All right. So I'm going to just skip this part showing that three-dimensional imaging. I want to spend time explaining that. So this device we call it black silver because even though it's a silver film, almost 100 nanometer thick, but it shows as a black color because it has this random nanostructures beneath that silver layers. So it provides very low reflections. Okay, so the mirror effect is less substantial, and the substrate supports a broadband SPR, so it can always find some overlap with the laser excitation and the fluorescent emission wavelengths of the microscope. And metal enhanced fluorescence is only about 10 times compared to the smooth silver and larger than 40 times compared to glass. So this is what happens on the surface, basically. And the lifetime of fluorophore decrease on this nanostructure as well. So what we have done is we started culture cells. This is our original goal, right? We want to culture cell on the surface of this nanostructure and start doing conventional confocal, fluorescent confocal imaging of cells. So start to grow cells on the surface. Particularly, we want a cell to grow on the surface with the nanostructures. So this is a pattern surface apparently, and those area slightly lighter they are actually a smooth silver surface. But those darker areas are those areas with the nanostructures. So after 2 days culture, we started observing some cells start to crawling on top of these nanostructures, and that's when we start doing the actual imaging. Right. And this is the SEM picture showing the cells really growing on top of nanostructure, and this shows you the scale between the cell size and the actual nanostructure size. All right, so this is the confocal imaging result. What we did is we used fluorescence dye to label both the cytosol or cytoplasma and the cell membrane. And this slide shows you the cytosol or cytoplasma imaging. In this case, we label the calcium in the cytoplasma. And this fluorescence emission is green. So what we want to point out here is in this field of view, you will see multiple cells, right? So here is 1, 2, 3, 4, 5, 6. Okay, in this crop imaging, we have 6 cells, but these particular cells are, especially brighter, much brighter than the other cells. The reason why is basically sitting on this particular squared nanostructure areas. That's not a big difference. I mean, that's not a big surprise to us. On the surface, when z equals to 0 micron, that means this is really on the surface. You will have some fluorescent enhancement on the surface, so that's not a big surprise. But once we start doing z-axis sectioning in a confocal imaging, trying to elevate our focal plane and above the surface, we started seeing these cells, cell fluorescence, become even stronger relative to other cells. You see, this is a 5 micron above the surface, this is 10 micron above the surface, this is a 15 micron above the surface. It's almost get to the tip of the cell on the upper side, right? So we start seeing this green fluorescence even 15 micron above the surface. This cannot be explained by the near field, electromagnetic field enhancement effect anymore. So this is really due to the coupling of the cytoplasma into some sort of a resonance mode inside the cell cavity. Well, people have already published a paper using cell as a cavity to do a lasing. They demonstrate single cell laser already. So this is actually corroborate that kind of result. All right, so this is a cytosol imaging, and this is a movie showing the three-dimensional rendering of that confocal imaging of the cells. These particular cells are sitting on top of the nanostructure or nanoplasmonic device. All the other cells are not. But well, there's one thing I want to point out especially is no matter you see the fluorescent, or actually green fluorescence or not, you always see this blue fluorescence. This blue fluorescence is basically, it's called Hurst staining in a cell nucleus. Basically, it's actually staining the DNA or chromosome inside the cell, inside the nucleus. So this basically tells us this is really a field enhancement around the cell membrane. You don't have that much cell fluorescent enhancement inside the core of the cells. That is why you start seeing those blue, even when you have 5 micron above the surface, or even 10 micron above the surface a little bit, but not for the cytosols. So you start losing the green even at 5 micron distance. All right, so that's a cytoplasma imaging. Here, this slide is showing the cell membrane imaging. We think is even more interesting. In this case, we label the cell membrane using this Invitrogen FM143FX dye to basically label the cell membrane, especially lipid part, and then use the same Hurst staining to label the DNA in a nuclei. So the DNA shows as blue color, and the cell membrane shows as red color in this case. Okay, so here is very, very clear, three-dimensional or in a perspective angle confocal imaging of cells. And this is one cell that you can see very clearly, especially its membrane structures. But all the other cells here in the same field of view, you can somehow see the nuclei, right? They're all blue, but not the membrane that much. So this, further support our hypothesis. This coupling between SPR to WGM is really happening on the cell membrane. It's really a membrane-supported resonance mode in the cells. So this shows that if we elevate our focal plane above the surface, start seeing only this cell showing up in this image above the surface. And then we did some quantifications about to find out how many times this fluorescent enhancement has been done by this particular configurations. And we show that especially for the maximum, for the focal plane with the maximum enhancement, our enhancement is almost 70%. It's very close to 100% already. Okay, so how do we use this? Well, this slide shows some control experiment. Control experiment is showing that this enhancement is not due to the silicon nanostructure itself. It's not due to glass. It's only due to you have this silver-coated nanoplasmonic structures. All right, so this is the actual application slide, and this carries some true biological significance here. For those of you who are doing the cell imaging, especially fluorescent confocal imaging, you probably know that nowadays you often have to do transfection of cells to couple GFP, RFP, YFP with the protein you want to express in the cell, right? So that's what you commonly do. But another thing that nobody actually talk about is you usually have to wait 50 hours or longer to see a significant expression level of the protein on the cell membrane or a cytosol. You cannot do the actual imaging right after transfection, or not even 12 hours right after, 12 hours after transfection. You have to wait for a few days in many cases to see the expressions. And in my view, and also in our collaborative view, is if you do this, you start losing some information happening in that period of time. So what we want to help them is, can we do this kind of imaging using the nanoplasmonic coupled WGM enhancement, so then you can reduce the time before you can do imaging, right? So this shows that we can get very distant confocal imaging of this particular paxillin proteins expression on the cell membrane only 12 hours after transfection. I believe this is quite an achievement because this can be applied to any kinds of GIP, RFP coupled protein expression and really help biologists to discover the lost information happening in the first 12 hours after transfection. Okay. By the way, the actual paxillin is a cytoskeleton proteins related to the cell motion and membrane properties of cells. And so to have this higher enhancement, higher sensitivity in the fluorescent imaging, not only helping you to be able to image in the early stage protein expression, but also allow you to capture those really fast events. Because when you have low sensitivity, you really have to increase your integration time for each pixels. So then if you want to get a full frame imaging of the whole cells, that takes longer time, right? And then if you have a low scanning rate, you're going to lose some fast occurring events like this calcium wave inside the cells. Calcium wave inside the cells, right? This is actually showing the calcium wave imaging inside the cells. So by increasing this intensity, you can then reduce the time to integrate at each pixels. So as a matter of fact, in this particular case, we actually use the shortest possible time for each pixels when we do confocal imaging. Okay, this is only 1.5 microseconds per each pixel. So the whole imaging only takes a couple of seconds to finish, basically. So then, with this kind of scanning speed, you can start to capture those fast occurring events like calcium waves. With that, I think I'm running out of time. And also I want to basically acknowledge our faculty collaborators, students, and our outside collaborators, and as well as fundings. So now it's 1 p.m. So I'm open to any questions. Thank you for your attention.
So we're out of the time, but I think we can afford 1 or 2 questions. So do you have any questions?
So what is the optical physics behind why having this plasmonic structure would cause the cell to become a resonant cavity? For the coherent light reflecting off the structure?
Well, cell can be a resonant cavity no matter it has this plasmonic structure or not. People have demonstrated using optical fiber couple these WGM resonance inside the cell cavity. People have already demonstrated. So the actual plasmonic substrate only serves as intermediator to couple the free space EM wave into the Swissman gathering mode resonance inside cells. Okay. It's just work as a fiber, optical fiber, but much easier, and also potentially can enhance the fluorescent intensity on the surface as well. Okay. All right. Thank you for the question. Yeah.
I see a very strong enhancement of the fluorescence from cells, from the nanostructure surfaces, and also above the surface. So when you do confocal, the resolution of confocal depends on the excitation volume.
Yes.
So the coupling will from the imaging surface, imaging plane.
Yeah,
is away from the surface.
That's actually very good questions. This is something we're also continuing investigating. Let me go back to this slide. This is a really good question. Yes. So in the confocal imaging, you basically only detect fluorescent emission from that focal volume. That's and not anything else. But your excitation light will not stop, will not be stopped at the focal volume. It will continue propagating, basically, onto this substrate. And so the actual substrate is constantly receiving some excitations, no matter where your focal volume is. And so once that happens, you always have some light that can be converted into plasma resonance on the substrate and then converted back into the resonance of the cavity. Okay. Yeah, so that actually, that is a very good question. That helps me to explain this kind of result, so that we don't have the uniform enhancement factor on any focal plane. This is actually variable, okay, variable. So we have optimum plane that has maximum enhancement. That is because you have different coupling efficiency. Because in the confocal, you only, as you said, only focus on, your light supposed only focus on the focal plane. You only detect fluorescent emission from the focal plane. Yeah.
And then another question is, when you cite your excitation plane away from the, like 20 or 10 micron away from the plasma surface,
Yeah.
then the question is there any degradation of resolution? Because some light will couple from the defocus region to the cellular, to the cellular structure.
I would say yes. You will have some influence from off focus in fluorescent emissions. Yes, you do have. We haven't looked at that particular aspect yet, because our focus at this moment is really on the sensitivity enhancement rather than resolution enhancement. Okay.
But there will be some enhancement in the kind of the wide field fluorescence.
Yes. So you will have some enhancement from the background as well. So the actual fluorescent you detect from that focal volume or focal point, it may have some influence, some interference from the background as well. That's the reality. So we're not claiming we're improving the resolution. We may even lose some resolution. But here, for the application, we don't care resolution that much. We just care about the expression level quantification. Then the sensitivity is most important to us. Okay, all right.
Oh, that's a great lecture.
Okay, thank you.
Thanks, Dr. Liu again.
Thank you.